Hey everyone, back again for the second micro lecture of the first day of class. What we're going to be talking about now is the history of microbial life on Earth, which is largely the history of life on Earth. So I'm going to start sharing some slides from chapter 1, and let's go. So these slides are available on the Camino site, so you can take notes on them there. What this is showing on the right is sort of the history, a timeline of life on Earth. What we know from our geologist and astronomer friends is that the Earth probably formed about 4.5 billion, maybe a little more than 4.5 billion years ago. Now, once the Earth had formed, it took a while for it to cool down enough for there to be liquid water on the surface. So that was happening here between 4.5 billion and 4 billion years ago. What we know, though, is that shortly after that, within like 3.8 billion years ago, life on Earth, the common ancestor of all that we see on Earth today, had appeared. So the evolution, what was going on in that period is speculative, but there's been some interesting research done in the last few decades on that. I'm going to mention real briefly on how we got to those first cells, and then we'll come back to this timeline towards the end of this micro lecture. So in that early sort of prebiotic stage, obviously the Earth didn't look like it did today. There was most of the surface of the Earth was still covered with water at that point. There was essentially no molecular oxygen in the atmosphere. The Earth was very hot still, so we have these hot oceans that had a bunch of prebiotic chemicals in them, things like methane and hydrogen and nitrogen gas, probably some ammonia, things like that. So how did we get from that prebiotic soup to the ancestor of all life on Earth as we know it now? Well, there's been some experiments done to try and simulate that early prebiotic period of Earth. The most famous one was done by a graduate student named Stanley Miller, working with his mentor, Professor Harold Urey, down in San Diego in the 1950s. And what Stanley Miller did was to try and set up an experimental apparatus to simulate this early prebiotic phase. And that's what's shown here. Here's what it actually looked like in the lab. But the bottom line is that there's several things you need to be aware of. One is that there was water here. This was the ocean, right? There's a source of energy here, right? So the Bunsen burner heating this thing up to simulate that hot early atmosphere. Then we also, of course, have a gas phase. There's gases in there like hydrogen and ammonia. And then up here, we have another source of energy, which is electrical discharge. This is simulating lightning in the early atmosphere, right? So essentially, you have the water here equilibrating with the atmosphere, getting heated up, running through here to running through here, being zapped with electricity, things condensing out of the gas phase, being put back into the water, et cetera, right? So the question is, what did they get? And what I'd like you to do is to take a look at that original Miller paper, and it's only like 2 pages long. It's on the Camino page, and I'm going to send you in the live class into breakout groups so you can take a look at it and answer some questions. Here's the questions I want you to look at. If you weren't there for the live discussion, then I want you to stop the recording here and take a look at that paper and think about these. What was Miller actually looking for in that prebiotic soup? What did he start with? And knowing what he found, if you wanted to take the experiment steps further, if you wanted to say, all right, what would we really need for a living system to evolve? What other molecules would you look for in that prebiotic soup that could have led to the first living systems evolving? All right. All right. So getting back to sort of stepping forward over the last few decades since Miller did his experiment, there's been a lot more learned. And this is just giving you a quick summary here of how we think or the trajectory that that early life took, right? So we started out, of course, with no life on that early Earth. We got our precursors in that prebiotic soup, and RNA was the first polymer that evolved. RNA, we know, has the ability to actually catalyze its own, both to catalyze enzymatic reactions and also to catalyze, in some ways, its own replication, right? So the theory is that self-replicating RNAs evolved, and they sort of joined up with lipid micelles, which could concentrate them and allow them to do their jobs more effectively. Obviously, there was no knowing this in advance. This is all abiotically. And so, that those primitive cells then, acquired the ability to actually catalyze the formation of peptide bonds. This is something that the 16S ribosomal RNA in bacteria is known to do now as part of the ribosome, is to help catalyze that peptide bond formation. Of course, most enzymes now are proteins, right? RNA can be an enzyme, but most of what we, the complex, highly specific, really fast enzymes are all proteins, right? So RNA was encoding the production of proteins, and then eventually that encoding job, or sort of the genetic repository job, shifted from RNA to DNA. And RNA, of course, still has a role, an essential role in replicating systems in living cells on Earth. But the functions that were originally with RNA have now been sort of shared out to proteins on the enzymatic side and DNA on the genetic side, right? So that's sort of the current theory as to how the first replicating systems evolved. Now, here's another thing I want you to think about. I want you to think about, what does it actually mean to be alive, right? What is a living system as we know it, right? Probably a lot of theoretical living systems, but I'm talking about, what does it mean to be alive in the sense that all cellular organisms on Earth share, right? So think about that. Think about what all those cells can do, and what would that first primitive ancestor of ours have to have been able to do in order for us to say it was alive, right? Think about that for a few minutes, and we're going to talk about that in class, and then come back. All right. Now, there was another major step that happened during life on Earth, actually more than one, but I'm just talking about this one for now, which is that pretty much all life on Earth, until recently, we thought fit into one of these two types. Either they were prokaryotic, which means they don't have a membrane-bound nucleus, or they were eukaryotic, which means they have their genetic material in a membrane-bound nucleus. And on top of that, they have other membrane-bound intracellular structures that we call organelles, okay? So we had prokaryotic cells and eukaryotic cells. And notice here it says up until the 1970s, that's what we thought. Well, in the 1970s, a guy named Carl Woese at the University of Illinois started studying ribosomal RNA genes as sort of a universal measuring stick for relationships between life on Earth, because all life on Earth has ribosomal RNA in it. So he started sequencing those not the same way we sequence DNA now. It's much more complicated. And it's talked about in your book in that mini paper in chapter 1, right? But the amazing thing that was recognized is that there wasn't just 2 types of life, prokaryotic and eukaryotic. There was actually 3 types, right? Based on clustering of relationships in these ribosomal RNA genes. One of them was the bacteria, which is most of what we had been calling prokaryotic life. But there was another kind of prokaryotic cells called archaea, which lived in really weird places, anaerobic sediments and hot springs, very, very strange places, hypersaline environments. But they look like prokaryotic cells, but in fact, they are separate from bacterial cells. And then over here, we have the eukaryotic cells, the ones with membrane-bound nuclei, right? And the really interesting thing is, by some measures, the archaea are as closely or even more closely related to the eukarya than they are to the bacteria. So these look like prokaryotic cells, but they actually are very distinct from each other. And we'll talk about those later in the class, okay? So let's get back to this timeline real quick, right? So origin of life, like let's say 3.8, 3.9 billion years ago. What we think now is that within a couple 100 million years, life had actually diverged into those 3 trajectories, or at least into 2 of those trajectories. And the archaea one, then through other events that happened, sort of diverged with what would become the eukaryote lineage, right? Now there's a bunch of other things that happened later as well, and I'll come back to the slide in just a second. But, well, actually, before I go on, let's just talk about them. Around 3 billion years ago, oxygen first appeared, and oxygen comes from photosynthesis that was at that point being carried out by bacteria. Now, we're going to talk about the evolution of organelles a little later in the class, but the plants that can carry out oxygenic photosynthesis essentially have resident protobacteria in them that we now call chloroplasts, right? So bacteria evolved the ability to produce oxygen from photosynthesis, which at its heart is just a way of getting your energy for metabolism from light, right? Now oxygen started to build up, and here we see about 2 billion years ago, there was a significant amount of oxygen in the atmosphere by that point. And that allowed aerobic respiration to evolve. And in eukaryotic cells, aerobic respiration is also being done by a sort of protobacteria that we now call mitochondria, right? So you see these endosymbiosis events that led to the origin of eukaryotic cells as we mostly know them now. And then those eukaryotic cells started to become multicellular organisms, a thing that bacteria largely haven't done, although there are social bacteria that work in sort of organized groups. But at any rate, the next big step then after the evolution of multicellular eukaryotes was the colonization of land. We'll talk more about this at some point as well. But that gets us closer to where we are now as land plants and land animals evolved. All right. Whew. How do we know these things actually happened? Well, there are some fossils, microbial fossils on Earth. The one that you see on your left here, these fossil cyanobacteria are the photosynthetic bacteria. So those aren't that old. Those are about 500 million years old. There have been a few fossils that are likely microbial in origin that are actually 3.5 billion years old. So way back in that early stage, before there was complex life, before there was oxygen in the atmosphere, etc. What do those look like? Well, they actually look like these things we call stromatolites, which you can see here on the contemporary Earth in Australia. These are basically colonies of bacteria that live in shallow water. Now, these obviously can live aerobically. You see they're exposed, but you can also find stromatolites under the water, and essentially there's sort of like microbial reefs in a sense. And we found things that look like these fossilized mats of microbial life. that are about 3.5 billion years old. There are very few places on Earth that actually have sediments that are that old because the Earth is dynamic, it's constantly recycling. But there you go. Last question, what about viruses? Are viruses living cells? Well, a little while ago, you guys talked about what it takes to be a living cell. So I'm going to leave it up to you to think about this. We're going to be talking about viruses in 2 more weeks. I'm not going to go through all this stuff now because I've been talking long enough for this micro lecture. And I am going to end this one now. Let's see. Stop sharing and end.